heavy. Humira do at least, around 22 billion per year sales, and overall life of the product, it did around more than 100 billion in the sales. So itself, it's a one company, just a one product, and it's been used in the multiple condition. Major is the rheumatic arthritis. Let's start with the drug development. Drug development, we're gonna cover what are the clinical phases goes in the drug development, what are the regulatory pathway for the approval, and different clinical endpoints. We will take as a example of the cancer drug development here, just to just to take a one indication. Uh, so, what is the cost involved for a drug development? Some says that it takes the 800 million estimate for a drug development, and majority of that is the cost related with the clinical development, which is around 72%. Um, the rest of the percent is the synthesis, preclinical, uh, and even some estimate that it takes more than a $2 billion, even for a one product development, depends upon the clinical condition, I believe. And if you look on the time frame, that how much time it takes from synthesis to the FDA approval, for any given anti-cancer drug, the average time frame is 16 years. It takes anywhere from 14 to 20 years. If you will granulate it, around 4.5 years for the preclinical, around 10 years for the clinical, and 1.4 year as a FDA evaluation when the FDA start reviewing the your new drug application submission. Other than the anti-cancer drug, usually 13.7 year is the medium time frame for the cardiovascular agent. Remember that it's a huge study. Any cardiovascular agent, the phase 3 studies, you're looking somewhere from the 5,000 patients. So this is a little bit a crowded slide. This is a flow chart for overall drug development. I took as the anti-cancer as the example. If you're gonna look the discovery and the lead optimization, um, start from identifying the target which is the target we're gonna target from the drug. It goes from finding the different analogs, doing the in vitro cell line study, then evaluate the compound to determine the molecular pharmacology and the target validation. Um, Once the lead analog do the structure activity relationship to identify that which side chain is more effective. These are the part of the discovery and the lead optimization. There are some parts are not related with the other clinical condition, like the NC1 cell screening result is not needed. Then once you identify the couple of lead molecules, it goes to the preclinical testing, where you have to first identify the acceptable animal formulation, and do the preliminary animal toxicity, like the assay biology, define the maximum tolerated dose. It's very important to define the MTD, that is the maximum tolerated dose, because that's where it's going to correlate to the starting point of the human dosing when you're going to the first to the human dose. Then development of a bioanalytical method. Mostly it's LCMS or ELISA. LCMS is for the small molecule, ELISA is for the biologics. Then you'd characterize the preclinical pharmacokinetics, like the ADME, T-half, AUC. These are important, and even you want to understand that how the enzymes are metabolizing the drug, because that's where the pharmacogenetics evaluation is very important, because every enzyme cohorts in the individuals are different, and that's what's going to differ the dose response relationship during the human studies. Determine if the compound is renally eliminated or not. These are the example during the preclinical testing. Then once you are identify all those things, you identify the carcinogenicity, cytotoxicity of the drug, then you go and do a formal toxicity study for the FDA. That's the GLP requirement. Depend upon the condition is chronic or the acute condition. the number of months of the studies varies. The systemic or the, if the drug is given by the topical, you need to go and do dermal tox studies. For the chronic studies, it goes up to the 9 months of the dermal tox study. Every day you have to dose it, repeat dose study on the mini pig model. And then once you establish the preclinical test, the product is ready to start a discussion with the FDA. And that's where you start doing the formulation development for the human formulation. And then that's where the clinical testing starts, which is the most expensive part. You go and do a pre-IND meeting, clinical evaluation with the FDA. And based on the negotiation with the FDA, you run your phase one trial, and then phase two, phase three, and the NDA submission. We're going to discuss more details in the clinical part in the next few slides. So overall success rate, right? So cancer drug overall success rate is pretty low. Only the 5% of the drug which enter in the phase one ultimately reach for the approval. Especially the blood conditions are the highest success rate, around 26%. But if you exclude the cancer drug, which made up to 31% of all the clinical trials right now going in the worldwide, overall success rate is 11%. And most of the drug fails in the phase two. There are the couple of reasons. The phase two, either the it's fail in the study, either in the efficacy or in the toxicity point of view, or the lack of funding. Because even if you look, the 70% of the drug development is the clinical. Out of that, I will say that 50% is just phase three, which are the bigger studies. Once you complete the phase three, 85% of drug when submitted to the FDA for their review, get approval. And because mostly you met with the FDA multiple times, you negotiate with them on the study design. Uh, that's where that they handheld you the clinical studies and make sure that we succeed. Drug with the rare condition, and that's where the Grace Therapeutics, I work mainly on the rare conditions and the orphan condition, where the total number of patients per year is less than 200,000. Most of the conditions does not have any therapy approved in the global space. But here, the barrier is a little bit low. Success is high because your clinical trials are smaller, but it doesn't mean that FDA compromised in the clinic or the safety. 58% of the drug that begins in the phase three basically get ended up to submission to the FDA, and finally achieve the approval. So if let's talk about the clinical phases. The phase one trial, which is very important because that's where you expose the drug first time to the humans based on the animal model. Um, the whole point is that what should be the starting dose. The starting dose is for the cancer drug is 10% of the LD50, especially in the rodent. And the phase one is so important for identifying the MTD, which we call the maximum tolerance dose. What is the maximum tolerance dose? Where we start seeing the side effect. So you price your side effect, that what kind of side effects are possible, especially in the oncology setting. DLT is very important. That is the dose limiting toxicity, that at what kind of the toxicity, because again you are looking as the risk benefit. There are the certain mild side effects, but if you are getting a more benefit, you take that chances. And then pharmacokinetics and pharmacodynamics evaluation at the phase one trial. um, the most therapeutic classes, for the other indication, the all phase one studies are done on the healthy normal volunteers. Only the oncology where you do in the patient, because most of the oncology drugs are cytotoxic in nature. You don't want to expose the healthy volunteers with the cytotoxic drug, because most of the drugs are not selective between the cancerian cell and the good cell. And then also, when you select the patients for the phase one on the oncology, you want to make sure that those patients are not getting benefited with the other chemotherapy, and that's where goes in the phase one trial for the oncology. And that's where it makes it a little difficult, to recruit the patients for the phase one trial on the oncology. Move on to the phase two trial where you start determining the efficacy in the different tumor cells. You want to know if my drug is working or not. You do more pharmacokinetics data on the patient cohorts. These studies are single arm typically. It means that you don't have a placebo arm. You can do a dose identification in the different arms, but typically you don't have a placebo arm here. The single institution means you have only the one center where you run the study because you want to be a more controlled environment on the study. And then you maximize the chance of detecting clinical response or biological activity because of using a single institution. Everybody practice the same way of the dosing. You don't have different nursing staff or the different principal investigators. In the phase two trials, again, it's a phase design, and basically we call the phase 2A to B, looking for one response at least in 12 to 15 patients. If you get a one response, you expand the study to the 30 to 40 patient. Ideally, the thumb rule is that if you don't see a single response in the first 12 patient, there is only a 10% chance that you rejected a drug, you not took to the phase three, which can give you a more than a 20% response rate, which is supposed to be the successful, in the phase three. But you take your chances because you don't want to fail in the bigger study, which is more expensive end of the day. There is no standard formula to go no go because the phase two study is not a placebo control study. It's not powered enough to get a statistical valuation. It's more on the scientific approach. The product goes further for the phase three trials. Phase three trials, which are large, hundreds of patients. In the cardiovascular, as I mentioned, it goes to the thousands of patients. Diabetic, it goes thousands of the patients because you need to have the right group of the population which is representing the study. It's randomized. Randomized it means randomization in terms of the demography, randomization in terms of the gender, randomization in terms of the placebo arm and the active arm. It's a multi-institutional study because you can't run a study in a single institute unless until it's a very rare condition where you don't have a lot of patient population. Multi-institutional remove the biasness from the study, and it helps you to complete the study faster because you can increase the recruitment of the patient in the study. And then it also helps you to file the application to a different regulatory body during with the one study. Like you can do the patients in the Japan, you can do in Europe, you can do in the US, and you can submit your application at the same time to all these regulatory body. uh, Needs to meet the clinical endpoint to submit the application for the review. Control group usually receive the standard of care plus placebo. Standard of care means that whatever the therapy is used for the standard at this point for that disease, and the broad selection of the patient representative community because you need them for a better demography in the study. Typical endpoint of the study for the FDA. This is more based on the oncology again, because I have to take a one clinical condition to present here. The TTP is the time to progression and the progression-free survival, PF. This is one of the endpoint FDA look. Overall survival is the gold standard in the oncology. You look for the overall survival, relief of symptoms, and the delay of events can be another endpoint. Delay of event like a death is a considered as an event here in the oncology. This is one slide on the orphan status. Orphan status, especially in the US, if it is fewer than 200 people year affects by a given condition, it's fall under the orphan condition. Once you get a product approved in the orphan condition, you get a market exclusivity up to the 7 years. Which is the most exclusivity you can get in the US. The reason is that because it's difficult to recover the cost of development because of the less prevalence. Now we will go to the once the product is done on the phase 3, you file the product to the FDA. What are the regulatory pathway? What are the approval pathway? There are the regular approval, which is you complete your phase 3 safety study and you did the direct evidence of the clinical benefit, like overall survival and reduction in the symptoms, or the other is the improvement in established surrogate clinical benefit. Basically indirect measurement for a clinical benefit. You identify that how much tumor is shrinked by the imaging technology can be considered as a surrogate endpoint. But end of the day, you need to go and prove the FDA that it works by overall survival in the phase 4 clinical trial commitment. Um, Accelerated approval is FDA is motivated to do that because of the life-threatening condition. There is no approved product for that indication. That makes FDA that okay, if they see the indirect benefit, they can go and approve. But it's a tentative, it's a conditional approval. You need to go further. You need to go and in the phase 4 bigger study while the patients are getting the drug. So phase 4 trial, it's a post-marketing period for, it's also expands to use the market indication that if the drug is only approved for a one indication, I mean to say one subgroup of a cancer, it can go to the another indication or another subgroup of the cancer, or rather than from the second line treatment to go to the first line treatment in the oncology can be done with the phase 4. Required for the drugs that receive accelerated approval, because accelerated approval is the conditional approval. You need to go whatever the time frame needed, either 3 years, 4 years, 5 years to submit your phase 4 data. You have to submit. FDA gonna review. They might grant you the approval, or based on the data, they might deny the approval, and you have to pull out from the market. So once you complete the study, it's all about translating the clinical trial result into the clinical care. I mean to say that's the real world, right? So a very good publication came out from that Tanak and colleague, which they compared the overall survival for docetaxel combination with the prednisolone in the prostate cancer. What they did, the same principle, these guys ran the clinical study, but what they did, they compared the 314 routine patients there with the 43 enrolled subjects was treated with the docetaxel and the prednisolone. And if you look, the overall survival for the routine patients, it's just 13.6 months. But when in the clinical trial, it came out to 20.4 months. Later, the TAX 327, the published paper came out in the New England Journal, and the overall survival was reported 19.2, which is nowhere closer to the routine patient. The reason is that the clinical study is done in the inclusion exclusion criteria for the patients. You exclude a lot of patients based on the criteria. So even if they look from the eligibility criteria from the based on the protocol, still for the routine patient it was 16.7, still nowhere close from the 20.4. So that's a concern with the FDA, that sometimes the product get approved based on surrogate model, but it does not work in the actual world. This is again the success rate slide. The oncology is 13.4%, and that oncology also small molecule is 14.3, large molecule is 11.5. Um, Very interesting that if you're going for the second and third indication, the product failed in the first indication, very small chance that it's going to be passed in the second or the third indication. Very recent example is um, if you look the remdesivir right now approved for the COVID-19, it was failed in the Ebola virus when they did the trial 6-7 years back. So basically first indication was failed, but in this case, the remdesivir pill passed for the second indication trial they did. Very interesting example. Four main reasons for a drug failure is the lack of efficacy. There is no, you can't succeed if the side effect uh, is risk versus benefit. If you can't, if the risk is higher than the benefit in the side effect, the product cannot be get approved. Pharmacogenetics is that if the subject variability is high, if cytochrome P450 or some metabolites, majority of the humans are in the different, in the different genogroup of the enzymes, the products cannot be approved. Pharmacokinetics, if the low biology, low half-life, protein binding, and action, but these are the low chances of the failure. End of the day, you have to prove the lack of efficacy. The efficacy needs to be proven, even the biology is low. That's ends the drug development part. We now going to the clinical pharmacology. The three principles of pharmacology established is the in the 16th century. What is the three basic principles? That each disease has a specific cause for which there is a specific remedy. Each natural remedy has identifiable component which is responsible for its efficacy. Look at the marijuana has more than 400 chemicals. Out of that, CBD and THC, which is being identified or for a pharmacological activity right now. The size of the dose determines the degree of response. That is the drug response relationship, dose response relationship. I mean, I will run these slides a little quicker because I'm going to run out of the time. Parculus, he was the Swiss physician, father of toxicology, known as. He established that dose makes the poison, which brings the concept of distinguishing between the pharmacology from the toxicology. Dose response relationship, very well said that there is all substances are poison. There is none which is not a poison. The right differentiate the poison from the remedy. Drug action, pharmacology versus the toxicology. Pharmacology is the interaction of the chemical and the macromolecules with biological systems to yield the therapeutic or the beneficial effect. Toxicology is the field of science that help us understand the harmful effect of the chemical substance either to the people, animal, or the environment, even to the environment. So it needs to be environment friendly. This slide I want to go through for a reason that the side effect, of course, the most of the side effect we consider that it's not good for the humans, but even if it is benefit, but it was not intended in the study, it's still considered a side effect. And it's a very interesting example, very successful drug is the sildenafil, which was synthesized by the Pfizer for the hypertension or angina pectoris. That was the primary indication. They ran the clinical study, but it was not efficacious in the bigger study. But in the phase one, they figured it out. There is a side effect, the mark erection, and that's where it finally approved by the FDA on the indication ED. National Cancer Institute defines the side effects in the 5 criterias. The grade 1 is the mild, moderate, severe, threatening, and disabling, where the neurological disabling, and the grade 5th is the death-related AE. I'm going to present very quickly that how it's been quantified in the clinical trials. So this is a crowded slide, but I'm gonna just want you guys to look at the ALT here, quantitative assessment, which is a liver function. So look at the grade 1, which is the 2.5 of the upper limit of normal, goes up to the 20 upper limit of normal, and then 5th is considered as a death. This is the severity of how you grade the AE in the clinical trial. If you have a qualitative assessment, if you look the cystitis, it depends upon the asymptomatic is the grade 1, then the severe bleeding, and then death is the grade 5. It's just why you do the grading, because you're going to look from the risk versus benefit assessment at the end of the day. What is my risk, what is my benefit, and that's where the product makes sense. The drug with a narrow therapeutic window, very important for the anti-cancer drug. Of course, the minimum effective concentration is the 3, which needs to achieve no matter what. This is more responsible side effect. But end of the day, in the anti-cancer drug, you really push the drug, dose in the plasma up to this level. The reason is that most of the anti-cancer drug is not that effective in the real world at the lower. To increase the chances to win the clinical trial, you really need to push the boundary up to the toxicity levels. Drug exposure effect relationship. You dose the patient by any route, either injectable, oral, or the transdermal. Drug needs to go to the absorption, distribution, metabolism, elimination. That's the pharmacokinetics, drug disposition in the other terminology. And then it's a pharmacodynamics, drug effects. That's the tissue effect, toxicity, efficacy, or the biological effect. But the genetic variation plays a huge role in terms of, even if the drug is in the stream, it doesn't mean it's gonna show the effect, because of the different genetic makeups of the humans or the different individual. And that needs to be studied well during the clinical trials, or even in the preclinical stage, at what is the understanding on the drug with a genetic variation. Let's go to the pharmacodynamics. That's where the clinical pharmacology comes in. Um, most drug works via receptor. That's we all know. And the drug receptor and drug target is cellular macromolecules, or the molecular complex with which the drug interacts and give effect as a cellular response. And commonly the drug alter the rate or the magnitude of the response, intrinsic cellular response, rather than create a new response. It's 99% of the drugs do that, unless until on the ulceration or the intestinal ulceration, the drugs works topically, does not interact with any receptor. It just works topically, it just covers the area, to reduce the it's like a band-aid on the wound, but it's on the intestinal surface. Drug receptor binding. What is the drug receptor binding? It's like a lock and key model, we know, but it's an occupancy theory. The response of a tissue of a drug or the ligand is a function of the number of receptors bounds to the drug. Again, the number of receptors bounds to the drug is very important, because the amount of receptors going to be engaged by the drug, the more efficacy it will be in the most of the cases. The site on the receptor where the drug binds is the binding site. The concentration of the drug is the important factor to the extent of the receptor binding. Typical needs to reach a threshold. That's where the threshold is very important, because that's where, look in the pain condition, if the consider the mu receptor, if the mu receptor is not engaged, certain numbers, the threshold is not going to reach, and it's not going to increase to tolerate the pain. Different kind of the drugs, agonist drug, and when we're going to talk about agonist drug is always a positive in the response. The drug which binds to the physiological receptor and makes the effect of the endogenous signaling compound. Uh, Primary agonist is a drug that binds to the same site where the endogenous lesion bind, and the allosteric and agonist is the drug which binds to the different region, but still it gives still mimic the same effect as a primary agonist. Antagonist is a drug which is basically the block and reduce. It's compete for the same site, or it can be a non-competitive for the same site, and it can be a functional also, which indirectly reduce the effect, physiological effect. Let's take an example of androgen receptor. This is for the prostate cancer. Testosterone goes in, 5-alpha reductase basically metabolize the testosterone to the DHT, dihydroxyl testosterone, which basically go and bind with the androgen receptor AR, which gets stabilized by the heat stroke proteins, and that goes inside the nucleus, and then it's eventually express the protein. So to break this cycle, you need a androgenous receptor right here, as agonist drugs. So there are the three antagonist drugs are, synthesized, flutamide and biglutamide and nilutamide. But these are non-selective and not very effective. So eventually, one of the analog got approved by the FDA, that is the enzalutamide. And if you look the panel A, the overall survival, and it's very interesting because if you look the other therapy, this is where 50% is on the around 13 month. And if you look, from here, with the drug, it is around 19 months. 6 months of the survival is changed, and amazing statistical value. The p-value is less than 0.001, and that's where you win the study. So that's the just example. There is a partial agonist. It's a minimal response, even it's bind with the same number of receptor as the agonist drugs. What are the drug properties you look during the development? Specifically, you look for the hydrophobicity, ionization state, conformation, and stereochemistry, S and R form. If you look the example of warfarin, the S is 4 times more potent than the R interaction for the vitamin K exposed reductant binder. I will give you one example of thalidomide. It's a very tragic incident happened in 1960s. More than 10,000 defected kids born in the Europe, primarily in the Europe, Canada, and Australia, even by the exposing a single dose of thalidomide for the pregnant woman. Um, um, this drug was that time taken for the sleep aid. So if you look the stereochemistry, the R is responsible for the effect, and S was responsible for the teratogenic side effect. That time clinical studies was not involved to do the study on the pregnant woman or for the teratogenic effect. But that's what we learn now. And if the clinical study does not involve the teratogenicity study, on the product label, you will get the label saying that this drug is not should not be used in during the pregnancy or during the lactating nursing mothers. The receptors property. Receptor property, basically, the hydrophobic and the hydrophilic part, interior and the exterior of the protein. And of course, how you see it. Fold is very important because that's where the configuration of the drug works in, and it goes sits inside that. We're going to see example in a couple of slides. Drug receptor bonds. We know this, the four types of the bonds: the van der Waal, hydrogen ionic covalent, from the bond strength. Usually, sometimes, more than one kind of the bonding happens. It's some relatively weak interaction, which creates a strong interaction. That's where we call the high affinity of the drug for receptor. Let's take example, Imatinib, a huge successful drug for the multiple myeloma. It's a kinase inhibitor. If you look on the panel A, that how the drug sits in the configuration on the folding of the kinase inhibitor. And that's where here is the van der Waal interaction in the panel B. And then in the panel C, you see the Imatinib is inhibiting or prevent phosphorylation of a activation loop of the kinase inhibitor, which prevents the catalytic activity. Uh, it's a huge successful drug published in the New Journal England of the Medicine, for the leukemia. Even in the phase 2 trials, it shows the very good results. Look at the Kaplan-Meier estimate for the Imatinib. We can see that even at the 12-month, the difference is 80% versus 30% for the other combined chemotherapy. And then you look at the basically, at the 24-month, the separation. You can see that imatinib is clearly separated from the standard of care in terms of the overall, cytogenic response with a huge p-value. Next comes the drug-receptor interaction. Basically, four kind of the interaction. It can be affinity, intrinsic activity, selectivity, and the numbers. Numbers are the number of receptors are available for binding. Selectivity is that the intended target, it should be a selective or non-selective. We're gonna see an example later on. Affinity, that how well the drug binds. So look at the selective example. It's androgenic receptor listed blue agonist, and then yellow is in the antagonist. Again, it depends, what kind of the target we want, alpha or the beta, or beta 1, beta 2, beta 3. Different drugs are selective or the non-selective. If you look on the non-amphetamine is non-selective even between the alpha and beta. Uh, if you look the Metoprolol, which is a very successful drug for the cardiac blood pressure management, it's a very selective for the beta, and that also just for the beta 1. So these are the tools you need to apply during the clinical pharmacology or drug development at the very early stage, that what is my target. Sweet binding, basically this is it changed the basically the structure, enhance affinity of the drug to the receptor. This goes beyond the lock and key model, what is the well known. The example is the insulin, but I'm going to give you the better example for the oncology, especially the formation of the platinum DNA adduct. Platinum is a huge successful drug for the oncology too, do the connect with the DNA sequencing, and that's where it's inhibiting the further the multiplication or the activation of the DNA because of the creating this adducts. So rational drug designing. What is the rational drug designing? Let me see how much time I have. More 15 minutes. Rational drug designing is the application of the structure activity relationship during the development of new drug. This structure activity relationship started back in the early 90s. The first was patented by the Bayer for the sleeping aid. I'm going back to the thalidomide example. We're gonna work out on this case study now. The thalidomide, if you will look, thalidomide works on the T cells, works on the COX for the inflammatory cascade, cytokines, and T and F even. So it works on the multiple, and of course we know the angiogenesis. So it works on the multiple receptor. The whole point is that can we develop an analog which can be a more selective rather than non-selective to all these receptors. We went for the zebrafish. Actually, NIH did this study for the angiogenesis. They identify more than 300 analogs, and they was looking for that the is that vessel length is overgrown, or the number of vessels get overgrown. The less number of the vessel number and the less number of the vessel growth is the high anti inhibitor for that analog. And then for the anti-inflammatory activity also, that how much neutrophil migrate towards the injury. And these are all the analogs they look, and then what are the amounts that get migrate. uh, Eventually they went and identify from the more than 315 analog, that 16 has anti-angiogenic property, 10 has anti-inflammatory, and 3 has both. They took all these all 16, 10, and 3 analogs for further genograph NC160 in silico model evaluation to identify the lead molecule which they want to take it for the animal studies or the human studies. But this tells that what they able to do with this. They able to establish the SAR for thalidomide analog. They know that which functional group alteration can be a selective or not selective for any given the receptors or the target. This is how I wanted to just present an example for thalidomide, that how the rational drug designing needs to be done. And that's the end of my presentation. But eventually the thalidomide, I want to go for the another slide, that two analogs got approved by the FDA, lenalidomide and the pomalidomide, both for the oncology indication. Thank you. So if you guys have any questions, it's most welcome. Hello. Dr. Sarvanan, did you get any questions, that you want me to answer?
Sir, 1 minute.
Yeah.
Sir, this is for a discussion, panel discussion. Anyone of delegate would like to for this topic? Please ask any one of the participant.
I think we have more 10 minutes.
Sir, good evening. Hello.
Hello. Yeah.
Hello. Sir, good evening. This is Pradeep from Vageshwari College and faculty of the college. Sir, do we have any selection criteria for the dose selection in the phase 1 going for the clinical trials? How do we generally have the criteria of selection of a dose for a clinical trial?
So initially, if it is a new molecule, which has never been exposed to the human, initially you have to identify the dose in the animal model, and that's done by the SAD and MAD study we call, the single ascending dose, multiple ascending dose, and then you look for the maximum tolerance. So there is a very clear guidance on the ICH, that how what dose needs to be exposed to the human. You have to identify, we call NOL. NOL, what is that? That is the minimum dose where it starts in a uh, uh, and then there is a conversion table in that guideline, that whichever animal model you choose, and what would be the conversion factor for the human based on the body weight and based on the body surface area.
Okay, sir. So do we have any specific regulation for that, sir?
Yeah, we do have. So there is guidance on the in the ICH. If you will look the on the guidance, you can get. I can give you the specific guidance name later on. It's not on top of my head, but if you will search for a non-clinical toxicological study, you can do that for ICH. And
Okay,
especially
thank
for
you.
oncology, it's supposed to be 10% of the LD50 on the rodent. So that's the starting dose for the oncology.
Thank you, sir. One more thing, sir. Like as we were discussing about the thalidomide, so when we see about the we say rational drug design, how do we generally, if at all, we have a configurational change, like as you have explained about the R and S specificity of the configuration in the thalidomide. So, if at all we
Yeah.
have a new molecule with different configurations, which one of is it effective? How we can really identify that case?
So it's again goes back to the discovery that first of all that if it is a resin administered with RNS and we see the effect and we see the side effects also, right? And we do it under an assumption, if unless until you do have a good structure activity relationship establishment, that which orientation works how. But in the absence of SAR, what you have to do, first of all, that can you synthesize a single isomer or not. If you can't synthesize, it's a muted point, but you're able to synthesize, then you can run in the clinical, not animal model, animal model, basically a disease animal model. You can identify that which one is has a more pharmacological benefit versus side effect. In most of the cases, the racemate mixture, maybe the both are effective. It's just the different fold. Might be the R is better active towards the S for the receptor. So again, it depends upon the can we synthesize, how well we know the SAR, and can we stabilize a single isomer.
Okay, so thank you. And if we have any adverse effects, can we say rule out with the help of CAD and QS or in any new moiety or novel identified molecules?
So you can do that, and that's where is the life cycle management. So what happened that again it goes to the risk versus benefit, right? So even if there is a risk, but there is a huge benefit, and you don't want to put a lot of money in your discovery or the new clinical study, new analog. Yeah, you go in the market with the existing product, but you keep on working on that if you can minimize the side effect with a new analog. Uh, you do the studies, you file the product with the FDA, you get approval on, better labeling, and then you discontinue the existing product. So again, it depends. It's a business decision that either you want to invest in the new analog to reduce the side effects or a trial, or you want to work on the new NCE. It's a business decision end of the day from the industry point of view.
Okay, sir. Thank you, sir. Thank you for the presentation. It was really very informative. Sir. Thank you.
Thank you.
Sir, one more question in the chat room. If any drug is not eligible for the clinical trial 3, in the sense if it is a fails in the clinical trial 3, can we go for the first clinical trial for the same drug, or we can continue, or we can stop the clinical trial?
So you so it's a why the clinical trial failed. That's the first question a company have to ask to the team, that what is the reason. Is my clinical study design was not appropriate, or it was not powered well. So again, it depends upon why it failed, right? So it can be failed because I fell in the borderline stats. So I supposed to get less than 0.05, and right now I am on the zero point zero 0.06. Do I need to power up the study, if I'm going to increase the the patient subject, like the number of subject in the study by 200? That's enough powered the study. Yeah, you can go for a next phase 3 trial. So that's always happened in our world. But again goes to the business decision, right? Because there is always a competition, and then some other companies running a trial with a different analog, and they are about to approve, how much market you're going to do. It makes sense for you or not, which is more effective, their product or our product. So it's a race, always. So it depends upon that. Hello. Hello.
Sir, wait, sir, 1 minute.
Yeah.
Mic on చేయండి.
Very big thanks for all participants.
Please, anybody? Any participant?
Excuse me, sir. Sir,
Yeah.
Please, Manasa.
sir, even after performing the all safety and efficacy studies in the clinical trials and estimating the maximum tolerance dose, why do we observe some unwanted effects, sir, after releasing into the market?
So yes, so of course, you have to do the post marketing surveillance. So that's what we call the pharmacovigilance. Uh, uh, doesn't matter how safe is the drug. You have to report any AEs reported in the from the as a market complaint needs to be reported to the FDA. It's always been put metrics to identify that what kind of the side effects is being reported post marketing, and the annually the labels of every drug modified based on the pharmacovigilance, post marketing surveillance side effects. And if it's a very serious side effect, drugs are being pulled out from the market based on the serious side effects. Especially the most common is the hypersensitivity, because of either of any excipients which plays the role in the hypersensitivity, and then that product needs to be pulled out from the market, and that needs to be reformulated.
Hello, good evening, sir.
Mhm.
Hello,
Yeah.
hello, good evening, sir. Sir, if we perform any another studies about already existing drug, which will be need any permission from the anyone, sir? Is it require any permission from anyone?
So, if you're talking about repurposed drug, which is already been
Yes,
approved
sir.
in the any places, and going by a regulatory pathway called 505(b)(2) pathway, uh, if it is a new indication and you are seeking for approval from the FDA, yes, you need to go and discuss with the FDA. You need to submit a IND, investigational new drug application, for this new indication, but only the people who are not required to file any regulatory asking from the bodies are the physicians. Physicians can administer any approved drug for any indication based on their evaluation, but industry can't do that. They need to use the FDA.
Okay, thank you, sir. Thank you, sir.
Okay. So big thanks for all participants for your active participation. Um, um, and thanks for Dr. C.H. Suresh, sir, principal of one of the Pharmacy College Nizamabad, and Dr. C.H. Pradeep, and Dr. K. Rajeshwar, and Dr. Harihom Sharma, sir. So thanks for your active participation. Let me remind you one of the famous quotation of the loyal Jack Lehman: never stop learning, for when we stop learning, we stop growing. Stay home, stay safe. Have a good day. Stay tuned for the next announcement. Over to the Jan, sir.
Thank you.
Thank you so much, sir.
Thanks a lot, sir. We hope the
Thank you.
session was insightful, informative, enlightening, and an academic repast. We solicit the gracious participation, cooperation, and support of our delegates in making the event successful. Kindly fill the feedback form using the link shared during the session in order to obtain the e-certificate. Do like, subscribe, share, and comment our official YouTube channel for upcoming webinars and to serve you better. For any queries, you can DM us at our email ID or feel free to call us on the contact number provided in the program flyer. Thanks a lot to the speaker, Mr. Amresh Kumar sir, Director of Grace Therapeutics from the United States of America, the Chief Patron, Dr. G. Srinivas Reddy Garu, and the convener, Dr. R. Ramakrishna sir, the Principal of Akeshwari Institute of Pharmaceutical Sciences, for organizing such a beautiful event. Last but not least, thanks a ton to Mr. V. Sridhar sir for his untiring and relentless efforts in making the event memorable. Good night, Shubharatri, stay home, stay safe, Jai Bharat.
Thank you.
Thank you, sir.
A small announcement to the registered participants. Hello. Small announcements to the registered participants. So for generation of e-certificate is available in and which is also available in the box of YouTube. And please go to the feedback link, and after submission of feedback form, you will receive one link to your registered mail ID, and with help of that link, you can download your e-certificate. And thank you very much for your active participation.